I'm Dr. Mike Cowley, and I'm a member of both the genetics and toxicology programs here at North Carolina State University. So my lab is interested in understanding the interactions between epigenetics and our environment. So I think most people are familiar with this idea that our genetic information is encoded within our DNA, and that our DNA is important for instructing cells on how to behave. But here's the conundrum. Almost every cell type within our body contains the same set of genetic information and the same instructions on how to behave. Yet clearly we're composed of many different cell types that have very specialized functions, from skin cells that keep us watertight and keep out bacterial infections, through to neurons that help us make decisions and store memories. So how can one set of genetic information be used in so many different ways? And the answer lies in how the cell interacts with or interprets the information that's encoded within the DNA. So for example, by controlling how a transcription factor might come in and bind to the DNA. When this system goes wrong, this can result in cell dysfunction or disease. So for example, many cancers can be partially caused by genes being inappropriately activated at the wrong time or in the wrong cell type. So we need to make sure that genes are switched on and switched off in the right places and at the right time. And this is what epigenetics is all about. It's the study of how genes get switched on or switched off. So what are these epigenetic mechanisms? These are structural or chemical changes to DNA that don't actually change the underlying genetic sequence, but influence how the cell interacts or reads the DNA. Perhaps the most well-studied epigenetic modification is methylation, which occurs mostly on cytosine residues in mammals. And for example, when a promoter of a gene is heavily methylated, this normally results in the gene being shut down or switched off. I mentioned that my lab studies epigenetics and the environment. So where does the environment come into this? Well, it turns out that epigenetic modifications can be readily influenced by extrinsic factors and environmental factors. And this could be anything from the time of day that your body is experiencing through to the amount of alcohol that you consumed last night. And these are important biological changes which modulate your metabolism so that you can deal with these environmental influences. But there are some environmental factors which are not quite so friendly. And these can induce epigenetic changes that persist over a very long period of time and can alter gene expression over a long period of time, ultimately contributing to disease. Now, one example that my lab is really interested in is the effects of exposure to the toxic metal cadmium. Now, this is particularly interesting and important to us because we know that there are populations of people, both within the United States and across the globe, who are exposed to high levels of cadmium, perhaps through their diet, or their drinking water, or maybe just in the soil in their local environment. My lab has shown that exposure to cadmium in people can cause widespread epigenetic changes across the whole genome, but these are particularly enriched at genes involved in metabolic and cardiovascular disorders. And so we're really interested now in trying to understand what are the mechanisms through which cadmium can influence the epigenetic modifications of these genes, and then what are the potential impacts for these changes on our long-term health and disease state. I wonder how this works in real life.